We're continuing our online lab journey with the chemical composition of cells, namely the macromolecules. So we know that organisms, living things, are made of cells, but we haven't really talked about what are cells made of. Well, in essence, you are what you eat. The food that you consume is going to provide these basic building blocks for you to use in your body to build your cells. Food products contain three main things: carbohydrates, proteins, and lipids. There is a fourth category called nucleic acids, which you should also be familiar with for lecture. However, we don't talk about them in the sense of food, since anything that was alive has nucleic acids in high amounts. So we're going to focus on the carbohydrates, the proteins, and the lipids for this lab. Let's go into proteins first. Keep note that the video here is not necessarily going to go in the order of your lab, so you'll want to pay attention there. Proteins are really one of the biggest building blocks we have in the body, and we use them for a wide variety of functions in all the different parts of our body. They really do pretty much everything we need, or a high amount of what we need within the body. They can help us transport things, they can help protect us from pathogens, they can function as signals or hormones as well. Amino acids are the building blocks of proteins. We call those monomers, mono meaning one. So these are the single units, the individual building blocks of a protein. And when we start linking these amino acids together, we start forming what we call a peptide, or a peptide chain. When amino acids are linked together, we call the bond between each amino acid a peptide bond. So you'll notice with the terminology, we're saying peptide a lot. If you see peptide, you should immediately think something to do with proteins or amino acids. So amino acids are linked together with peptide bonds, and when we form long chains of these amino acids that have been bonded together, we call that a polypeptide. Poly meaning many, and peptide, amino acids. Some proteins are specialized, and we call those enzymes. These are specialized proteins that help speed up reactions without actually being used in the reaction itself. A lot of chemical reactions that we require to happen in our body will take days or even weeks to happen if we let them happen just as they are. With an enzyme, we can speed that up from days to weeks to seconds and minutes. It's absolutely essential that we have enzymes to help speed up these reactions. Otherwise, we would not be able to break down the food that we eat. We would not be able to help maintain homeostasis in our body. We would really lose the ability to control a lot of things happening inside of our bodies. Let's look at the amino acid structure here. So there are 20 different amino acid structures, and this is our main building block. This is what we're using to build our proteins. We have an amino group, a carboxyl group, also known as an acid group, and the R group, called the side chain. The amino acid structure has some parts that are going to be maintained no matter what kind of amino acid you are, and some parts that can change depending on which amino acid you are. Every single amino acid has the same basic backbone of the amino group and the acid group. So everything we see here that I'm kind of circling in is going to be the same for every amino acid. The only part that varies from one type of amino acid to another is the R group, also called the side chain or the variable group. So this is what makes each amino acid, in a way you could say, unique. You're going to be watching a video about testing for proteins. Proteins can be detected by the reagent called the Biuret reagent. When there's no protein present and we add Biuret reagent, the reagent will stay a blue color, kind of a pale sky blue-like color. But if protein is present, the reagent will turn a violet or purple-like color. You'll be watching a video demonstrating this procedure. Make sure to understand what's happening, as you may be asked questions about this procedure on your lab exam. Our next category is the carbohydrates. Carbohydrates can vary from very simple to very, very complex. Simple ones include things like sucrose or glucose, whereas complex carbohydrates include things like starch and glycogen. When we're looking at carbohydrates, we probably think of sugars. Sugars are just one group of carbohydrates, and they tend to be very simple. They tend to be on the sweeter side too. So you think of like table sugar, which would be sucrose, is a glucose and fructose stuck together. But not every carbohydrate is sweet, especially when you start making complex carbs, which are these long chains of sugars glued together. Things like starch. Starch does not necessarily taste sweet. If you bite into a potato, you're not going to get this sugar rush, right? It's not going to taste really, really sweet to you. We have the ability to store this glucose, to store these carbohydrates away for later. This is one of the reasons why that we don't need to constantly eat, because we have the capability of storing these things for later use. We can store them as a glycogen, which is a complex carbohydrate, which has these long chains of glucose molecules all stuck together. Glycogen is a polysaccharide. When we see saccharide, we should think something to do with a sugar. Think like if something is saccharin, it tastes sweet. So a polysaccharide, poly meaning many, saccharide meaning sugars, many sugars would be a complex carbohydrate. Disaccharides and monosaccharides are simple carbohydrates. Mono meaning one, and di meaning two. So monosaccharides are a single sugar, disaccharides are two sugars. So between the two groups, you're 1 or 2 sugars in size. That's pretty small. The polysaccharides are where we get these really long, crazy chains of tens, hundreds, thousands of sugars glued together. So let's look at some of these. So here we see some of the monosaccharides, like glucose and fructose and galactose, which are all like this one single ring. These are all types of sugars. And the polysaccharide, where we see these glucose molecules that have been linked together with chemical bonds. And so we look at it, we zoom out, we see this long chain, and then it's branching off to the sides. There's several, many sugars to create the entire molecule called starch. You're going to be watching some videos testing for 2 types of carbohydrates, the simple carbs and the complex carbs. For the simple carbs, you'll be watching a video on the Benedict's reagent and how we can use it to test for those simple carbohydrates. If there are no simple carbohydrates present and we use the Benedict's reagent, the reagent will stay blue. Again, kind of that sky blue, that pale blue look. If simple carbs are present, then the reagent will vary from green if there's a low concentration of sugar to red if there's a very high concentration of sugar. So we can kind of think of it like a rainbow from blue to red. Blue meaning there's no sugar, green being low, yellow and orange meaning there's a medium amount, and red meaning there's a lot of carbohydrates, a lot of sugars. So you'll watch a video demonstrating the procedure as well as the results of the Benedict's reagent test. For complex carbs, you'll be using iodine to detect starch. Iodine, if there is no starch present, will keep its yellow, golden, amber kind of color. Iodine by itself is relatively yellow, and if it does not change from its yellow, there is no starch there. If starch is present, then a very drastic color change will happen, where the iodine will turn blue to black, very deep, very dark in color. It's a very strong contrast compared to the normal yellow coloring of iodine. So that's another thing you'll be watching a video on, watching the procedure, as well as understanding what some results mean. Again, for both of these, you'll need to watch those videos and understand the procedures, as you may be asked questions about these on that lab exam. Lipids are our last group. Lipids are insoluble in water, meaning they don't dissolve in water, and we can call that hydrophobic. Things like fats and oils, steroids, cholesterol, and phospholipids are all within the lipids category. The lipids category is really broad. It's a really wide variety of molecules held within this category. The one thing that unites all of them, though, is their insolubility in water, that they do not mix well with water. When we're looking at the triglyceride, which is the standard fat, they have a very basic kind of framework. 1 glycerol, which we see here in red, and 3 fatty acid tails, which we have demonstrated in this blue-purple color. Things like steroids and phospholipids will vary a little bit from this, but do be familiar with the triglyceride. We'll talk more about steroids and cholesterol and phospholipids in later labs. So you might be wondering, why don't the lipids and water mix very well? Well, it turns out that water is polar, meaning that the molecules, or I should say the atoms that make up water, have very slight charges on them. Oxygen has a very slight negative charge, and the hydrogens in water have a very slight positive charge. Whereas with lipids, they're nonpolar, meaning they don't have these partial charges at all. Because of this difference in whether or not they have these very slight charges, it means lipids are repelled by water. They're hydrophobic. They don't want to mix well with water because of the difference between polar and nonpolar substances. This is why a lot of times when you are making a salad and you pull that bottle of salad dressing from your fridge, you have to give it a good shake before you pour it out on your salad. Otherwise, you'll get a bunch of oil or a bunch of water and not much else. Because they don't mix well and they separate over time, by you shaking it, you're kind of redistributing the oil throughout the water. But if you set that dressing on your counter and give it a few minutes, you'll start to see it separate. I recommend maybe doing that with the dressing you might have in your fridge right now. If you have some like Italian dressing or balsamic vinegar dressing, give it a shake, watch it separate. You're seeing that hydrophobic nature of lipids in action. Now you're probably wondering, well, I know I need fats in my diet, and I know that I'm made up of a lot of water in my cells, so how the heck does that work if they don't actually like each other and mix? Well, there's a process we call emulsification. Sometimes we do need to mix lipids and water in our body, and in order to do that, we need an emulsifier. So something like bile salt. Bile salts, which is an emulsifier, will help kind of combine these hydrophilic and hydrophobic components. They can help mix together molecules that are polar or nonpolar. And the reason why they're so good at that is because they have parts that are polar and they have parts that are nonpolar. So here we have this big kind of bubble of fat here, and we want to break this down into smaller droplets so that we can use it within the body. The bile salts, they have a yellow portion here that's hydrophobic and the green portion here that is hydrophilic. When they combine with this big ball of fat, the hydrophobic parts will face the fat and the hydrophilic parts will face the water. And so we can break this large globule down into smaller droplets that are easier for the body to handle because we have this very special molecule that's both hydrophobic as well as hydrophilic. Hydrophobic meaning water-fearing and hydrophilic meaning water-loving.